In this lesson, I want to discuss one of the most important synapses that we will discuss in Human Anatomy and Physiology 1 lecture. And this one is called the neuromuscular junction. Neuromuscular junction. Neuromuscular because we have a neuron and a muscle cell. So the neuromuscular junction, or NMJ. The neuromuscular junction is one of the most important synapses that we deal with, and is the model synapse for synaptic activity. So what is a synapse? And a synapse is a pretty simple thing. It is nothing more. We have a neuron communicating with another cell. And neurons can communicate with a muscle cell, they can communicate with other neurons, or they can communicate with a gland. So this one, neuromuscular junction, very obvious, very clear that it is a neuron to a muscle. There is a space between the two cells, a gap called a synaptic cleft. If the two cells touched, we lose the efficiency of the action of the synapse, and most likely would not get binding of neurotransmitters. So when these two guys go to communicate, they use chemical messengers known as neurotransmitters. The neuron, known as the presynaptic cell, possesses neurotransmitters, and we're going to draw them in. And we have presynaptic vesicles in here that we have contained neurotransmitters, the red dots. And neurotransmitters are chemical messengers who are used to communicate between two cells. The particular one here is called acetylcholine. Acetylcholine is the neurotransmitter in the neuromuscular junction. So I'm going to call it ACH. Acetylcholine is the red material here in the neuromuscular junction. Now let me erase our NMJ. Let me just write it up here for you guys, NMJ. Now, there's no, on the postsynaptic cell, we have an area here, and I'm going to use red again just to highlight this. An area of the postsynaptic cell, its membrane on the muscles called the motor end plate that I have in red. Now the motor end plate of a muscle possesses receptor proteins here called nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors because they will bind to nicotine as well as acetylcholine. Acetylcholine, though, once we are set, we are pretty much ready to get this muscle and get ready to show you guys the whole process. We got receptors and we got transmitters. Now, before we do anything, and I'm going to draw it outside the neuromuscular junction to set this muscle in resting, but we have a protein that we need to look at, which is an active transport protein, and I'm going to draw it as a little blue oval. This is the sodium potassium ATPase pump. And the job of the sodium potassium ATPase pump is when I take ATP, for every molecule of ATP, we break it down into ADP plus an inorganic phosphate molecule. When we do this, the sodium potassium pump brings in 2 potassium cations and kicks out 3 sodium cations for every molecule of ATP expended. What this does, though, why I really hit this very hard, is that we have high K plus inside and low Na plus on the inside. But on the outside of the cell, sodium is high, potassium is low. And this will set up the whole entire process of passive forces that will drive the muscle. So sodium potassium ATPase pumps are responsible for resting of a muscle cell, resting. And this maintains it at about negative 95 millivolts. Now, the resting potential of this muscle is now set, and we're ready to begin doing some interesting physiology. Now, the very first thing that has to happen, we have to kind of think of my neuron here as the boss, and my muscle cell as an employee. Now, with this idea kind of going on here. We are ready to take this guy from resting and we're going to change him. So what we got to do first is that muscles only contract, only work when the nerve says to. So the neuron has to send a message. Now how does it do this? What we do is an action potential arrives on the cell membrane, and I'm drawing it like little lightning bolts on the cell membrane of the neuron, the presynaptic cell. When this happens, voltage-gated calcium channels open on the neuron, and calcium from the outside comes in, Ca2+. Calcium comes in and results in exocytosis neurotransmitters. So the neurotransmitters bind to the presynaptic cell's membrane to the terminal end, and this, because they are made of the same stuff, it results in exocytosis of acetylcholine. So this is exocytosis, the release of neurotransmitter. They come across the synaptic cleft, and it will bind to a nicotinic acetylcholine receptor. Now, when nicotinic acetylcholine receptors are bound to, this is when things get interesting. Now, we want to take it away from resting. So I'm going to erase this. Remember, potassium is in a high concentration, sodium is in a low. Inside the cell. Outside the cell, sodium is high. Now, I'm going to use purple when nicotinic acetylcholine receptors are bound to by acetylcholine. This results in opening of chemically gated or ligand-gated sodium channels. Now, we call them chemically gated because they are controlled by chemicals like acetylcholine. So when a chemical binds to it, so this is a drug, it either could open it or close it. And this is a ligand-gated or chemically gated ion channel. Now, the ions that this is for is sodium. Now, where do you think sodium is going to go? Now, we know sodium is in high concentration outside the cell, low in. So it is going to go, sodium will go into the cell. Na plus will flood in. This will make, now we got to think about something a little bit more. Remember the sodium-potassium pump? The sodium-potassium pump made my cell polarized. It polarized my cell, making it negative on the inside of the cell membrane and positive on the outside. Now, because of this, when sodium rushes in, the cell was polarized. And because it had opposite charge on each side of the membrane. As sodium comes in, it's more positive. Now, it comes in for two reasons. Number one, the cell was negative on the inside, and it's attracted by that negative charge, because it's positive and negative attract. Number two, and another very important reason, sodium is in a high concentration outside the cell, low in, simple diffusion, passive process. So it floods in. When this happens, my inside of my cell becomes more positive, and it is now depolarized. Now, a depolarized cell and a muscle cell does some really interesting things. So, for the instance for us, the sodium-potassium ATPase pump is not doing anything for us right now. Once all that sodium comes in, the sodium-potassium pump, it's no longer doing its job. Now, I'm going to erase all the physiology on the presynaptic cell just to clear things up and to show you guys what happens during the depolarization. Now, depolarization, sodium rushes in, and some really interesting things happen. Now, we do have extensions of the cell membrane of a muscle cell called the sarcolemma, called the triad. And what the triad is one T-tubule and two terminal cisternae. Now, when the muscle cell depolarizes, an action potential generates on the cell membrane of the muscle cell, on the sarcolemma. And this dumps down into the T-tubules. Now, the terminal cisternae are storing calcium, so when the action potential arrives, this results in the stored calcium dumping out of my terminal cisternae, Ca2+. And when this calcium leaves, This results in what we call excitation, contraction, coupling. And you can't read, it's very hard to write that quickly on our excitation, contraction, coupling. This makes a muscle cell contract. So the hard and fast rule for this is calcium is in a high concentration in the terminal cisternae, a muscle is relaxed. If calcium is in a low concentration in the terminal cisternae, the muscle is contracted thusly. So depolarization results in a muscle contraction. Now we need to rest this muscle. How do we go back? How do we get away from our depolarized state? Well, we need an enzyme. Now here I can use several different colors, but we get limited on colors. I usually like to use pink. Now I'm going to use a gray color here, and we have an enzyme that breaks down acetylcholine. It is acetylcholine esterase, ACHE, acetylcholine esterase. And it will break down acetylcholine from the nicotinic acetylcholine receptors, and now that they are clean, we no longer have acetylcholine bound. That automatically closes my ligand-gated sodium channels. So I no longer have chemically gated ion channels or ligand-gated ion channels open. No more sodium is coming in. But the cell is still positive, so how are we going to get it negative? Well, I'm going to use another, and it's always hard for us because we have a limited colors here with this, but I am going to use red this time because we no longer have acetylcholine inside my synapse. And let me go ahead and remove acetylcholine esterase. Now, using red, I'm going to draw in, because we have an action potential here, voltage-gated potassium channels open, and potassium leaves the cell, because potassium is in a high concentration inside the cell and low out. So K plus leaves. Now as this is also going on, acetylcholine, as it's broken down by acetylcholine esterase, the choline is recycled and acetic acid will go on to be used in cellular respiration. So those events are happening simultaneously. Now, as the potassium leaves the cell, it was depolarized. Let's repolarize it. Repolarization results from the opening of voltage-gated potassium channels. Potassium leaves. So what's going to happen is this cell goes back to being negative, but highly negative. And we don't want to be that negative. Now, these are voltage-gated. The voltage completely changes, so we are now going to lose that action potential, and potassium results in relaxation of the muscle. This is why if you have low potassium, you get cramps. Now, what then has to happen? The potassium leaves, the voltage drops, voltage-gated potassium channels close, then we end where we began. Sodium, potassium, ATPase pumps reset everything. 2K plus, 3 sodium plus for every molecule of ATP expended. We produce ATP plus inorganic phosphate. So ATPs, it is an ATPase because ATP changes conformation. This cell is now back to resting. And this is some of the most complex physiology that you guys will ever get. If you can understand this, you will understand anything in human anatomy and physiology. But this is critical for an understanding of both pharmacology and pathophysiology, which you guys will both take in your nursing programs or other advanced courses and other advanced learning programs. And this concludes my video on the events at the neuromuscular junction. Thank you.